ways to go in areas like simplifying sample prep and increasing read lengths for things like the microbiome. So there's continuous improvement that needs to happen across the board here, and obviously price continually reminds me. But I think that the basic fundamentals of the tool
Anything you
set,
multiply by 100,000 makes a really big number.
really big numbers. But I think the fundamental tool sets today are so enabling compared to where we were just 5 years ago. And we were woefully underpowered 5 years ago in terms of tackling these problems. And I think today we have it at the disposal of people like Craig, the tools to go get this information and integrate it.
I think Jay's more right that people probably want to accept that the genetic code is actually going to be the predictive element. And people always say, what about epigenetics? And I want to issue t-shirts that say, epigenetics are still genetics. Everything that people measure as epigenetics originated with the genetic code. That's what we proved with the first synthetic cell. We just put in synthetic software and everything derived from that. And so ultimately, if the databases are big enough, we should be able to predict almost everything from the genetic code.
So something that I know is interesting to a lot of people in the room is how is NextGen sequencing going to affect drug development in the future?
Well, I mean, I think it's beginning to have a pretty big impact now. It's been used historically, in limited ways for drug development. Back in the 90s, the large pharma companies were all doing large-scale sequencing programs for target identification. I think you've seen over the past couple of years, after a lull, some resurgence in the use of sequencing by biotech and pharma companies. Regeneron announced recently they're going to sequence 100,000 exomes, which is kind of a shocking thing to hear them say. Decode in Iceland has imputed the sequence of most of the Icelandic population, and they were bought by Amgen for $450 million because of the database that they were able to create. And so I think in target ID and in enriching clinical trial populations, in beginning to sequence the clinical trials themselves, I think we're right on the cusp of that beginning in a serious way. It's actually been a real surprise to us that pharma didn't do this earlier, and from a scientific perspective, we could have been running microarrays on every patient in clinical trials 10 years ago. And when you really probe under the covers to figure out why that was, the reason was that they were afraid of creating data that they couldn't explain to the FDA. And I think that's been an underlying problem in the use in clinical trials. And I think that's the new FDA, if you will, is beginning to realize that NGS really does create that level of uncertainty, and they have to figure out how to live with it. And so I think because of that willingness to live with uncertainty of data you can't explain, pharma's beginning to adopt it again at large scale.
But it was also fear of market segmentation. So
Back then it was.
Back then.
No, no, it's not.
So the Pfizer drug that works on these AOK mutations in lung cancer, the clinical trial failed. If you knew that it worked on that subpopulation, you could have done a very small clinical trial very quickly. And so what's changed is the definition of a blockbuster. A blockbuster used to be a drug that was sold very broadly and therefore had broad market acceptance. And now a blockbuster is one where it costs $9,000 to $10,000 a dose for treating a small number of patients in these small genetic subpopulations. So that's what's finally, I think, driven it to be acceptable in the pharmaceutical industry to consider using genomics, and it should totally change how clinical trials are done, the speed and the cost, which means a lot more things can go through clinical trials and hopefully shorten the time for getting new drugs approved.
And success rates will hopefully
Yeah.
lower.
So let me ask you the same question about diagnostics.